So in today's lesson, we're going to be learning about alternate pathways for cellular respiration. Thus far, we've talked about how carbohydrates are broken down for energy, in particular glucose, and the various destination of the products of the breakdown of glucose and where they end up, and how they play a part in harnessing the chemical potential energy in that molecule in order to produce ATP. However, when we consume food, we know that various nutrient groups are present. So now it warrants a discussion of, well, what does happen to lipids and what does happen to proteins? Because we know that those are a part of the foods that we consume. How do those huge molecules get broken down, and how do we use the chemical potential energy in their molecules in order to produce ATP? So it should make sense to you that those are large molecules and that they would normally get broken down or digested into their component parts, their monomers. And so the monomers are going to enter cellular respiration at different stages, and that's what we're going to talk about today. Now, another part of the discussion is going to be, well, what happens to those organisms that are anaerobic? What happens in the absence of oxygen? How do those organisms harness energy from their foods? So let's first have a conversation around protein catabolism. But before I move on, I should mention to you that there is a bit of a hierarchy in terms of how foods are digested, right? So if we consume food, then our carbohydrates are going to be metabolized first, or catabolized first, I should say, right? So we have our carbs first. Then in the absence of carbohydrates, our body is then going to metabolize or catabolize the lipids in our system. And then as a last resort, our proteins are going to be catabolized. So if we're talking about the keto diet, for instance, the way that it operates is that if we eliminate carbohydrates, then what's going to happen? Well, in the absence of carbohydrates, our body is going to be metabolizing or catabolizing lipids. And so if carbohydrates aren't available and all that you've consumed are lipids, then you're going to see a reduction in your fat stores as well, as a result of your body trying to find ways to break down molecules to gain more ATP. Right, so we can talk about people who unfortunately have eating disorders. If they deprive themselves of food, then they're likely depriving themselves of carbohydrates and lipids and proteins. And in the absence of both of those, once your body depletes its stores of lipids and carbohydrates, you're going to see the protein in their body being catabolized, so hence muscle loss. And in particular, we need to be concerned about heart problems. Remember, the heart is one big giant muscle responsible for pumping blood throughout your body. So in the absence of that, people with eating disorders can suffer cardiac arrest, which is a big problem. All right. So let me get rid of this. I'm just going to do that. There we go. And let's get back to our conversation around protein catabolism. So proteins, when we break them down, we end up with amino acids. That should not be a big surprise, right? We can see here there's a peptide bond joining this first amino acid to the second amino acid in that sort of primary structure. And so our body's first job is going to hydrolyze that peptide bond by the addition of water. So one of the many reasons we consume water. So in that breakdown of that peptide bond, we yield an individual amino acid. So that would be the first process in breaking down proteins that we consume. Now, the next part of this is going to be a process called deamination. So deamination, just all that involves is removing the amino group. You can see it in the word there, deamination, amino group, from our amino acids. So, of course, all amino acids have that central carbon, they have that amino group, and they have the carboxyl group, and then that variable R side chain, right, that basically provides us with the identity of the individual amino acid in terms of which of the 20 that it represents. So that amino acid is going to be removed in a process we call deamination. So why does that matter? Well, ammonia, or that amino group, is a waste product and it's toxic to us. So our body needs to find a way to get rid of it. We can't really metabolize it for energy. So what happens to it? Well, it's released as waste, okay, as ammonia, and that gets excreted in our urine. So let's look at what remains after that. So after that, we have, well, I'm just going to highlight it here. After that, we're left with basically a 2 to 3 carbon compound, right? So we know that if we have only a 2 carbon compound, well, what other 2 carbon compound do we know about in cellular respiration? Well, what comes to mind for me is an acetyl coenzyme A. And so if it's an acetyl coenzyme A, then where is it going to end up? So let me write some of that down. So, oopsies. So if it's acetyl CoA, then we know it's going to go into the Krebs cycle, right? However, the problem lies in what this variable R chain is here. So if it's a variable R chain that contains a lot of other carbons, then depending on what it is, it may enter at different stages of cellular respiration. Yeah, here's my other junk. Let's get rid of that. Okay, so that's it for amino acids, really. We can't really say more at this stage. It's beyond the scope of this course what happens to each amino acid based on that R group. But at some point, if you continue on in your studies in biology, that will be part of your conversation. All right, next I want to talk about lipid catabolism. So when we consume lipids, they're first going to get broken down into their component parts. Now we're specifically going to talk about triglycerides, which, you know, are the primary type of lipid that we would be consuming. So as you would expect, those triglycerides are going to get broken down into glycerol and fatty acids. So let's take a look at this. So here just to remind you about that process, right? Here's our glycerol and here are our 3 fatty acids. We're going to hydrolyze that in order to break down our triglyceride into its component parts. And what results is that glycerol. Now glycerol is a sugar, really. It's just a sugar. It's got those hydroxyl groups, which classifies it as a carbohydrate, but it's a 3-carbon carbohydrate. I'm going to talk about that in a minute. Now, our fatty acids, really, although there is that carboxyl group, we also have a bunch of carbons, and depending on the fatty acid, the number of carbon and hydrogen groups that we have is going to vary, but we definitely have a lot of carbon groups here. So let's first talk about the fate of our glycerol molecules. Now I tried sort of to highlight for you and remind you that it is indeed a sugar molecule, and maybe that prompted you to think, well, if I have one 3-carbon molecule and then I have another 3-carbon molecule, then perhaps in some way they might get converted into glucose. And that's sort of what happens, and that process is called gluconeogenesis, right? So genesis meaning like beginning or birth or whatever, neo meaning new, and then the gluco meaning sugar. So here we have our glycerol molecule that, you know, can eventually be, one other possibility is that it gets transformed into dihydroxyacetone phosphate, which we know as DHAP, right? And then so DHAP can therefore go on and just continue on in glycolysis, or, right, because it eventually gets converted into G3P. And so DHAP can also, however, be transformed into a glucose molecule and then start glycolysis from the beginning. So that is the fate of our glycerol molecule in our. So I also tried to hint at what might be going on with our fatty acids. So by discussing how they really are just long carbon chains. So what ends up happening with our fatty acids is that they're converted into a bunch of 2-carbon acetyl coenzyme A groups. And that process is called beta oxidation. So, and those two, just basically breaking them down into little 2-carbon chunks, essentially. And you'll remember from our earlier conversation about when we compared carbon carbon bonds to carbon-hydrogen bonds, that we harness a whole lot more energy from carbon-carbon bonds, about twice. So if we compare a 12-carbon fatty acid, for instance, to two 6-carbon glucose molecules, so in essence we're saying here the number of carbon atoms are the same, then despite the fact that we have the same number of carbons, the type of bond that we see in each of these individual molecules is going to be different because of the presence of oxygen in our glucose. And so the fatty acids are going to yield us about twice as much energy. So that's why we often refer to fatty foods as being very calorie dense. So there we have it. That is what happens to the protein and to the lipid that we consume. So this is a really good overview. I like this diagram because it kind of sums up all of what we've been talking about. Our new textbook doesn't really talk about nucleic acids so much, but you'll remember that nucleic acids have nitrogenous bases. So any molecule with a nitrogen in it is going to have ammonia as a breakdown product. Our proteins, we talked about, first metabolize or catabolize into amino acids. They are deaminated, and then the ammonia leaves. But depending on what results, we might end up with, 3 carbon pieces, 2 carbon pieces, maybe some other pieces, that are a little bit larger, might end up being one of the components in the Krebs cycle. So that's one possibility. We know what happens. Oh, we didn't talk about polysaccharides, but I think we sort of may have discussed this when we talked about glucose. If we have a polysaccharide, say for instance like a starch molecule, really all a starch is a polymer of glucose. So those glucose molecules are going to be, we're going to get those from the breakdown of that polysaccharide, like an amylose or so on. Okay, and then there's our sort of regular aerobic cellular respiration process. Lipids, they're first going to get broken down, and this of course if we're talking about triglycerides, they're going to get broken down into glycerol and fatty acids. The glycerol is going to become glucose by that process of gluconeogenesis. Fatty acids are going to, by a process of beta oxidation, become acetyl coenzyme A molecules and then go through the whole process of aerobics of the Krebs cycle. There it is. Okay, so now we need to talk about, what happens in the absence of oxygen? So anaerobic respiration is what occurs. So you'll remember that in stage 4, so when I say stage 4, what am I talking about here? Stage 4, I'm talking about the electron transport chain primarily. And that electron transport chain, we use that in order to, first of all, harness the energy in those electron carriers, NADH and FADH2, and then that's used subsequently in chemiosmosis. But another purpose of that electron transport chain is to regenerate NAD plus and FAD so that they can go back to the very beginning of aerobic cellular respiration and then go through the whole process again. However, in the absence of oxygen, which is what aerobic respiration is, there needs to be another way to recycle these electron carriers. And also there needs to be a way for organisms to get whatever energy that they can. So that process is called fermentation, and there are a couple different types of fermentation. We're going to look at ethanol or alcohol fermentation and lactate or lactic acid fermentation. So let's take a look at ethanol fermentation. I want to just pause for a moment. I want you to take a look at this diagram and see if you can figure out what is going on, just as an exercise, just to kind of try to figure out what the heck is going on here. All right, so now what I want to do is I want to talk to you about fermentation just sort of in general. I want to remind you that very early on we talked about how glucose is our starting material, and that how glycolysis, the process of breaking down that glucose, happens in the cytoplasm, and that actually is an anaerobic process. No oxygen is required. So in other words, regardless of whether we're talking about aerobic or anaerobic cellular respiration, glycolysis is going to be a stage. Glycolysis happens whether we're talking about aerobic or anaerobic respiration. So in glycolysis, you'll remember that we end up producing a net gain of 2 ATP. So just envision that here you have, I'm going to say, for example, a yeast molecule, and a yeast undergoes anaerobic respiration in the absence of oxygen, and so it will definitely undergo glycolysis. So just like any sort of normal aerobic respiration process, glycolysis is going to occur. We're going to yield 2 ATP and the 2 NADH. Now, the problem begins with what happens to the 2-pyruvate that are generated from glycolysis in the absence of oxygen. So normally, during aerobic cellular respiration, well, what would happen to those 2-pyruvate? Those 2-pyruvate would go on to be oxidized into acetyl coenzyme A. But in the absence of oxygen, that simply can't happen. So what happens instead? Well, it's decarboxylated, right? We remove a carbon dioxide, but instead, in the absence of oxygen, we don't produce an acetyl coenzyme A, we produce acetaldehyde. So acetaldehyde is a 2-carbon compound, just like acetyl coenzyme A, but it's what occurs in the absence of oxygen. It's what's produced in the absence of oxygen. Now remember that one of the reasons that this process is occurring is because we have this NADH right here that was produced during glycolysis, and we need to be able to regenerate it back into NAD+. So the way that happens is that NADH has to get oxidized, and that happens by the reduction of acetaldehyde into ethanol. And so that is essentially why this process is referred to as ethanol or alcohol fermentation, right? It's this process of regenerating NADH into NAD+, right, by oxidizing it. And in its being oxidized, acetaldehyde is being reduced to ethanol, and that's our byproduct of the fermentation process. So let's look at the details here. So what is happening first? Carboxyl group of pyruvate is removed, right? So that decarboxylates it into acetaldehyde. The NAD+ needs to be regenerated, so acetaldehyde is going to be reduced by the NADH, and that produces ethanol. So which types of organisms are going to undergo this? Well, yeast and bacteria are going to undergo this process. But you can imagine that we're talking about a process that can occur in super complicated or complex organisms because we're not producing a lot of ATP, right? So in this process, only glycolysis is producing any ATP. So that's not going to be a whole lot of energy. It's not going to sustain all of the required activities that need to go on and be supported if we're talking about super complicated organisms. So there we have it. Okay, so same as we did in the last activity, the last component of this note, what I want you to do is I want you to take a look at this diagram that's representing lactate or lactic acid fermentation, and I want you to identify, well, what is the same here and what is different. Just take a minute to analyze that just for your own learning process. Just take a look at, well, what is the same and what is different? All right, so let's take a look. So for me, what I see that is the same is that, of course, we're starting with glucose, glycolysis is happening, and we're also producing ATP. It looks as though because glycolysis is occurring, we're producing NADH, and that eventually is going to need to get recycled back into NAD+. But what seems to be the same as well is that the pyruvate are a product of that glycolysis, but what is different is the fate of that pyruvate in the absence of oxygen. So remember that one of the reasons that the fermentation is happening is to regenerate our electron carrier by oxidizing NADH. So in the case of lactic acid fermentation, instead of pyruvate being converted into acetaldehyde, it's not decarboxylated at all. It is simply converted into lactate, and that's happening by its direct reduction into lactate. So why does that happen? Well, because NADH is being oxidized back into NAD+. So remember, in any kind of redox reaction, there's that coupling, that oxidation reduction happening. So if NADH is being oxidized, well, it has to be oxidized by something. It's oxidized by the fact that pyruvate is being reduced into lactate or lactic acid. So let's look at some of the details of that. Okay, so pyruvate, as we just saw, is reduced by NADH to form lactate instead. There is no carbon dioxide being released here, though, because lactate is a 3-carbon compound. Okay, so we're not removing the carboxyl group from our pyruvate. So where does this happen? I think most of you are aware of, particularly since many of you took exercise science, that lactate or lactic acid accumulation is a pretty painful process that happens when you are exercising pretty vigorously. So if you're exercising pretty vigorously, there may not be enough oxygen available to sustain aerobic respiration, but your body still needs to produce energy. When you're exercising, you're burning energy. Your body needs to find ways to break down molecules to keep providing you with energy. So, what your body's going to do in the absence of enough oxygen is undergo lactic acid fermentation, because even though you're not making 38 ATP as you would in aerobic cellular respiration, you're still making some ATP, just not very much. It's not super efficient. Okay, so your body's going to produce the lactic acid as a byproduct, and thereby get a little bit of ATP. So what's going to happen is that lactic acid is going to be in your body. It's going to cause some pain, but it's going to eventually be carried through your bloodstream to your liver. And by panting while you are exercising, you're helping to move your lactic acid around in your body. You're getting some oxygen going, so that will help you to create ATP via aerobic cellular respiration. But that lactic acid is going to end up in your liver. It's going to get converted back into pyruvate. The process by which that happens is you're not really responsible for. It's beyond the scope of this course. And if it is, of course, pyruvate at that point, it can go on and to be oxidized if there's enough oxygen available into acetyl coenzyme A, and you're going to see it enter the Krebs cycle and then our electron transport chain. So the panting that you're doing is going to provide that extra oxygen required to take that lactate and move it into being carbon dioxide and water. There you have it. So there's our quick breakdown of how molecules other than glucose are catabolized, and how our glucose is catabolized in the absence of oxygen. And so hopefully if you have any questions, you will reach out to me, and we can hopefully clarify any points of confusion, or if you have some additional curiosities.